Hi, in this video we are going to look at a process called translation. Now, in previous videos we talked about DNA and the structure of DNA, and we talked about DNA replication and how this takes place in the nucleus, and it makes photocopies of every single gene on DNA. So basically it's like taking a cookbook you find on the internet and you print out every single page, every single recipe, in order to have a copy of that cookbook. And that was DNA replication. And there were 3 steps. We unwound the DNA using DNA helicase. We copied each side of the DNA using DNA polymerase. And then we put our 2 cookbooks back together using DNA ligase. Well, in the case of protein synthesis, we have to do what's called transcription and translation. And in the case of transcription, transcription is how DNA is copied in order to make RNA. Now, in the case of transcription, transcription also happens in the nucleus. But when you need to make a protein, you don't need every single page in that cookbook. You just need the one recipe you need. So in the process called RNA transcription, we go to our DNA, we find the one gene, the one recipe we need, and we just photocopy, we print off, however you want to put it, just that one recipe. We don't need to carry the entire cookbook to the ribosome where the protein's getting made. We don't want to risk that cookbook getting damaged. We don't want our DNA to leave the nucleus. We don't want to risk our DNA getting mutated. So in another video, we talked about transcription. And transcription is how our bodies take DNA and use it to make RNA. So just remember we have this thing called a genetic code. And this is the sequence of nitrogen bases we have along the DNA. Number 1 here represents DNA. So it'll be like having 2 pages, or left side and right side. And so we have, just like having page 1 or page 2, it's 2 recipes we have here. And remember, every 3 letters on a piece of DNA is called a triplet. And every triplet tells me one ingredient that I need. And remember, the ingredients we're going to use are amino acids. So therefore, for example, GGA, that would be like a 3-letter word telling me, hey, you need this ingredient. And then TAC would be another 3-letter word that says, hey, you need this ingredient. But number 1 represents DNA, and DNA is the cookbook. So it has all the recipes that we could ever possibly need in our genetic makeup. Number 2 represents messenger RNA. When we need to make a protein, we only need the one recipe. I don't need every single recipe. I don't need to be carrying this 80,000 page cookbook back and forth. So I go to my grandmother's house and I take a picture of the recipe that I want, or I hand copy the one recipe I want, or I just go to the internet and I download the one recipe that I want. And this one recipe is what we call messenger RNA, because it's going to carry the message to the ribosome in order to make that protein. And in the process where we make RNA, this is called transcription. It also has 3 steps. We're still going to unwind DNA, but this time we're going to use an enzyme called RNA polymerase. That way DNA knows what it is it needs to do. If DNA helicase attaches, it's going to slowly unwind and slowly photocopy every single recipe. If RNA polymerase attaches, that's DNA's clue, oh, I only need to print out just one page. I only need one recipe. So RNA polymerase will unwind the DNA. Then an RNA polymerase will also photocopy, link everything up, A's and U's and G's and C's together. And then RNA ligase will come behind and put everything back together. So I have my piece of DNA and I have my photocopy. So when we talk about protein synthesis, we have these 2 parts. Transcription is how we use DNA to make messenger RNA or transfer RNA, RNA in general. We just always use messenger RNA as our example. So with transcription, those are those 3 steps where we're going to unwind DNA. It's like you go and you turn on your computer and you find the one recipe that you want. Then we're going to copy it. That would be like you printing off the one recipe. And then we're going to put everything back together. It'd be like you closing the cookbook. And this is going to occur in the nucleus, because now I just have that one photocopy. I have the messenger RNA. If messenger RNA gets messed up, it's not a big deal. If I take a picture with my phone and then I get home and I realize that it's blurry, not a big deal. I can take a picture again. Or if I print off a recipe from my printer and it smears, not a big deal. I can just print off a new copy. That's how messenger RNA works in our bodies. It's just this single recipe. And if it gets messed up, not a big deal, because we can just go to the original source, the DNA, and make a new copy. If DNA gets messed up, that's a big deal. So in transcription, we're going to use DNA to make this piece of RNA, any kind of RNA. We're just using messenger RNA as our example. Translation is going to happen at the ribosome. Transcription is at the nucleus. It's like me going to my great-grandmother's house and pulling her cookbook down from the shelf and opening it up and taking a picture of the one recipe that I want. Then I'm going to close her cookbook and I'm going to put it back where I got it. That cookbook is not going to leave the house because if it gets messed up, I know I am in trouble. Well, your body's the same way. DNA is not going to leave the nucleus because if it gets messed up, you are in trouble. Because remember, mutations aren't, a lot of times our body doesn't realize we have a mutation, and so that's going to cause that cell to be damaged or to die. So I've gone to my great-grandmother's house and I've taken a picture of the one recipe I need. Now I'm going to take that back to my house and I'm going to actually make the recipe. That's what happens in translation. Translation is where our cells take that one recipe to the ribosome. And the ribosome is the chef. The ribosome will read the recipe and put the ingredients together the way that they're supposed to. And remember, the ingredients we're going to be using are amino acids. So in the case of translation, this is where we're going to use RNA in order to make a protein. And this will occur at the ribosome. So in the case, as it says here, translation takes place at the ribosomes. And the main job is to read the messenger RNA. Just like you're reading a recipe, and the recipe tells you, okay, put this ingredient in the bowl, or put this ingredient in the bowl, put this ingredient in the bowl. This is what happens in translation. We have this recipe here. We have this piece of messenger RNA. And again, remember, we divided up into groups of threes. And these groups of threes are called codons. And what the ribosome is going to do is it's going to read this like a sentence. So it's going to translate what this says. And it'll tell me what ingredients that I need in what order in order to make my protein. So this is actually step one of what we call translation. Remember, there's 3 steps to everything. There's 3 letters in a triplet, there's 3 letters in a codon, there's 3 letters in an anticodon. There's 3 steps to making DNA. There's 3 differences between DNA and RNA. There's 3 kinds of RNA. There's 3 steps in making RNA. There's 3 steps in making a protein. The very first step of making a protein is called initiation. And you know that initiation means to start something. Well, that's what has to happen here. Okay? The messenger RNA has to get to the ribosome. So it'd be like you picking up the recipe in your hand, and the first thing any good chef does is get all of his ingredients. And remember, the job of our transfer RNA right here, that's our measuring cup. Our transfer RNA is going to carry the ingredients to the ribosome. Okay? And so think of your transfer RNA as being measuring cups, and they're holding ingredients. So if you're the chef, you're the ribosome, you're going to get your recipe, you're going to get all of your ingredients so that you're ready to start making the recipe. And that's step 1 in translation. Step 1 in translation is called initiation. This is where we're going to get everything ready and we're going to start making our protein. So again, transfer RNA is going to, my measuring cups, they're going to have all the amino acids laid out in front. And this is going to happen at the ribosomes. So what's happening in initiation, again, we have the messenger RNA leaves the nucleus and goes to the ribosome. So it's like you're taking the recipe from your grandmother's house to your kitchen, and you're the chef. And so you're holding that recipe in your hand and you have all the ingredients lined up in front of you. And you are ready to start making the protein. Well, after initiation comes what's called elongation. And just like the name says, we're going to make our proteins longer. So what's going to happen is the start codon of the messenger RNA attaches to the ribosome, and this is what's called initiation. At the same time, our transfer RNAs have all of our specific amino acids. So we have all of our measuring cups lined up in front of us. So 1 and 2 together are what we call initiation. Well, you're holding the recipe in your hand, and the recipe, remember, is a series of letters. So I'm going to go just write some letters up here, like I might have AAA, I might have GGU, I might have CCA, so on and so forth. And the next step is what's called elongation, and this is what 3 and 4 are going along with. So what will happen is the ribosome will yell out, hey, I've got AAA on my recipe. And the transfer RNA that matches that, its anticodon would be UUU. It says, oh, that's me. And it's going to dump its ingredient into the mixing bowl. The ribosome says thank you, and it moves down and yells out GGU. And the match to that, CCA, says, oh, that's my ingredient. And it adds its ingredient to the mixing bowl. And remember, we are adding amino acids with these things called peptide bonds. So we are elongating our protein. Well, eventually we're going to reach a stop codon, something like UGA. Just like every sentence has a capital letter, so you'll know that your sentence starts, every recipe starts with the same 3 letters, and that's what we call the start codon. Okay? And that start codon is AUG. That's how we know, okay, the recipe starts here. Well, just like you read a sentence to the end, and you know a sentence ends because it has a period, all of our recipes also have periods at the end of them, and these are what we call stop codons. So once the ribosome reaches the stop codon, it goes, oh, okay, I'm done with this recipe, and that's what we call termination. So again, 3 steps, 1 and 2 together are what we call initiation. Steps 3 and 4 together are what we call elongation, and then obviously at the end, we're going to have termination. So like I mentioned, the start codon is always AUG. So you can see here in this picture, this will be like initiation. We have AUG right here. So we're going to add our first ingredient. Then notice we're going to move to the second 3 letters. We're going to add the next ingredient. Then we move to the next 3 letters and we add the next ingredient. So that is elongation. You can see how I'm adding these amino acids together in order to make a protein. Again, this is just another picture of translation. You can see initiation, elongation, termination on the side. Initiation, again, when you initiate something, you start it. This is just where you're starting to make the recipe. So you've got your mixing bowl, which is the ribosome. You've got your recipe, which is the messenger RNA. You've got all of your ingredients in measuring cups. Those are your transfer RNA, and you're ready to start making your protein. Then you'll start reading the recipe, and you'll start putting things in your mixing bowl. That will be elongation, and eventually you reach the end of your recipe. You're done making your protein. That's termination. So again, in translation, remember each piece of messenger or each piece of transfer RNA carries a specific amino acid. 3 bases on the messenger RNA are called a codon, and 3 bases on the transfer RNA are called an anticodon. And make sure you know how to read the amino acid charts. Remember, there are 64 different possible combinations when you put these 3 letters together. And this is just another example of a codon chart. So you can pause this video at any point in time so that you can practice these codon charts. And then again, with transcription versus translation. Transcription is going to happen in the nucleus, and this is where we take DNA and we use it to make copies of RNA. Translation is going to happen in the ribosome, and this is where we take that RNA and use it to make a protein.